Hi, my name is Colby Ellsworth, and today we'll be talking about cellular communication, which is basically how all cells communicate with each other. The first topic we'll be discussing is the G protein pathway, which is the most common pathway used within cells. This pathway contains something called a G protein linked receptor. We'll just draw this right here. We'll just call this REC for receptor. This receptor can be found along the membrane and acts like the hand of a cell. Pretty much, when this receptor will change shape in order to fit the appropriate signal molecule called a ligand. Pretty much, this ligand is a molecule sent by another cell to this specific cell in order to send this cell a message to make a specific protein. And once this receptor changes shape, it allows phosphorylation to occur within the cell. This process, after this phosphorylation occurs, this process activates the G protein, which is pretty much like a relay point found within the cell. And this protein will receive the message from the receptor protein and take it to a specific enzyme or protein found along the membrane. Now, this will usually be GTP, and then this GTP is mostly used to turn on or off functions within the cell. It's usually functions with either in the nucleus or the cytoplasm. And these processes are mostly transcription or translation. And pretty much transcription is the making of messenger RNA, which is used to make a specific protein that this ligand calls for. So the signal is sent to maybe the nucleus, and eventually a new protein is made according to the ligand's instructions. Next, we'll be talking about the tyrosine kinase pathway. This pathway is pretty much involved in the growth and emergency repair of the cell. But it can also act like a catalyst that is used to activate multiple relay points, up to 6 at once, which are then used to carry out other processes in the cell. So pretty much what happens is that you have your pathway right here, and again, a ligand comes and it connects to this protein. What then happens is ATP is used to power this protein, and these little humps that are here, or little bumps that are on the sides, are like charging stations or activating stations. They activate these unactivated enzymes, or not enzymes, but proteins. Now, once these proteins connect and once they are charged, they then leave and go do their specific process which helps initiate the cell's response to the ligand's message. Now, pretty much, what a good way to think about this is that in order for a cell to maintain its structure, it constantly needs to repair itself. And this is a very efficient way for the cell to do so because of these 6 little ports that it has in it. It can charge up to 6 at a time, and multiple of these are found along the cell's membrane. Now, the structure equals repair thing is just an easy way to remember, because you always need to have processes going to keep the cell intact and alive, so that it can eventually lead to a split, and so more cells can be made. Next, we'll be talking about ion channel receptors, also known as ligand-gated ion channels. Pretty much, what these ligand-gated ion channels do, they're like control points for a certain signal. What they do is that they again accept a ligand and what this ligand does is that it allows the gate to open and allow ions, we'll call these, maybe sodium ions. It'll allow them to diffuse into the cell, which then allows it to depolarize the cell. Next, we'll be talking about intracellular receptors. Now, intra means to be within something. So, these receptors will not be found along the membrane, but inside the cell itself. So, we'll just put this receptor right here. Call it RAC for short again. Now, ligands will come into the cell and they will connect to these receptors. Now, once they do this, this receptor will diffuse into the nucleus of the cell and will pretty much set up these things called transcription factors. And these factors, start the making of mRNA. And this mRNA will be used to make other proteins within the cell. Finally, we'll be talking about secondary messengers. The first secondary messenger we'll be talking about is cyclic AMP, or CAMP for short. Pretty much how this is made is a adenylyl cyclase will combine with ATP and it will make cyclic AMP. Next, we'll talk about IP3. And pretty much what happens here is that IP3 will be made from a G protein that has a certain message from a ligand, and it will combine with PIP2, which is inactive, And once this G protein connects to PIP2, it will break this bond between the I and the P and turn it into IP3. Once this happens, the IP3 will travel to the endoplasmic reticulum, and will the endoplasmic reticulum is like a storage place for certain ions such as calcium. And to pretty much open up the ER, you need IP3. And once IP3 attaches to the ER, it binds with a receptor protein. So we'll do this. We'll put IP3 right here. And it will connect with this receptor protein on the ER. And once this receptor protein changes shape in order to fit the IP3 to it, this other secondary messenger called calmodulin will come out and attach to, we'll call it cal for now. It will attach to microfilaments and will then activate muscle contractions using the sliding filament theory.